Thank you for honoring my father, Dr. Saul Hertz, who conceived and brought from bench to bedside the medical uses of radioactive iodine, RAI. He'd been using stable iodine in X-rays as a non-surgical method to treat goiters, as surgery was dangerous and expensive. A eureka moment occurred on November 12, 1936, when Dr. Hertz spontaneously asked MIT President Carl Compton, could iodine be made radioactive artificially? Harvard funded the initial research. MIT Rad Lab director hired physicist Arthur Roberts, PhD, to work with Hertz. Roberts produced non-cyclotron I-128. Hertz and Roberts successfully demonstrated the use of RAI as a tracer to study thyroid physiology. The director of the Rad Lab did no work related to the research. It is documented that the director forced his name on the author's list once the paper had been approved for publication. Correspondence verifies the director had difficulty working with Hertz and Roberts as they were outsiders. Importantly, in 1937, at the time of the preclinical studies, Dr. Hertz conceived of RAI to treat thyroid carcinoma. How do I get the next slide? I don't know how to do it. Somebody's going to have to show me. Sorry, this is going to take
Just
a
the
while.
green button. Just
Okay,
the green
got it. Thank you. On March 31st, 1941, Dr. Hertz rushed over the Charles River Bridge with a flask of MIT-produced RAI. He administered the very first therapeutic use of an artificially produced radioactive substance. Uptake testing and dosimetry were used to determine a safe, effective dose. In 1942, he conducted and reported his trials of cancer patients with thyroid cancer patients to the Markle Foundation that funded the MIT cyclotron. By the way, it was $30,000 to buy a cyclotron in the day. Such a deal. In 1943, before leaving for World War II, Dr. Hertz advised New York Dr. Seidlin in the use of RAI to treat thyroid cancer. Patient BB arrived at Montefiore Hospital in New York and had had his thyroid surgically removed 15 years earlier. BB was treated with RAI as directed by Dr. Hertz. No new lesions appeared, and some completely disappeared. However, in 1952, BB succumbed to anaplastic carcinoma. I think I may have missed a slide here. Okay. There he is. That's BB. After the war, Hertz's military leave of absence was not honored at MGH. His replacement at MGH went to publish Hertz's work as his own, omitting both Hertz and Roberts. In 1946, with funding from the Navy, Dr. Hertz refined the use of RAI to diagnose and treat thyroid carcinoma at Boston's Beth Israel Hospital. Hertz stated, "My new research project is in cancer of the thyroid, which I believe holds the key to the larger problem of cancer in general." In September 1946, he founded the Radioactive Isotope Institute with facilities in Boston and New York. In 1946, in June, he's quoted as saying, "Demand is expected for radioactive iodine, and as research develops in the fields of cancer and leukemia, for other radioactive medicines." He successfully treated thyroid cancer and researched the use of radionuclides and hormones for other forms of cancer. A May 1949 Harvard Crimson headline read, "Hertz to use nuclear fission and cure for cancer." The cost of radioiodine was dramatically reduced with the production of RAI from the atomic pyres. In 1949, Hertz established the first nuclear medicine department at the Massachusetts Women's Hospital, reported as opening a new division where radioactive isotopes will be used to study and treat disease. Hertz stressed the tracer-targeted approach. With tenacity, courage, and passion, Saul Hertz overcame the racism of his time, a world war interrupting his work, the establishment pushing back, and the fear factor to bring his work to fruition. A cancer survivor emailed, "The cure delivered on the wings of prayer was Dr. Saul Hertz's discovery, the miracle of RAI. Few can equal such a wonderful and precious gift." Yes, our next speaker carries the legacy forward with more than 1,600 published papers, 1,400 patents, and is co-founder of Moderna. So please welcome MIT Institute Professor Robert Langer.
Well, thank you all for coming, and it's really an honor to be able to follow up Barbara and to really, you look back and you see the enormous impact of what Saul Hertz has done. I asked Barbara what she wanted me to say. She sent me this, I just took it right from her email. So it's how I've built on the foundation, which is a remarkable foundation. Training great people and establishing companies. So I'll just be very brief. My background, I got my degree in chemical engineering in 1974. And when all my friends went to work in the oil industry, I wasn't that excited about that. And I took a job actually at Boston Children's Hospital, by far the lowest paying job, but with a man named Judah Folkman, and who was a surgeon. And I was the only engineer in the entire place. But Dr. Folkman asked me to try to help him solve a problem, which was he had a theory that if you could stop blood vessels, that might be a whole new way to treat cancer someday. And he was widely criticized for that theory. But, and so we had to develop ways to study blood vessels. There wasn't any way to do that in 1974. And so the way we did it is there's certain sites that don't have any blood vessels, and we put tumors there, and then we had to create tiny little nanoparticles that could deliver large molecules, which had never been done before. And at that time, everyone said that that was scientifically impossible. Dr. Folkman and I hadn't really read the literature saying that, so we tried anyhow. And I actually worked on this for a long time. I found over 200 ways to get it to not work. But eventually, we did get it to work. We published 2 papers, one in the journal Nature in 1976, showing for the first time you could create tiny particles, like a nanoparticle, that could deliver large molecules, including even nucleic acids, which RNA and DNA are. And one in Science, also that same year, using this approach to actually isolate the very first angiogenesis inhibitors. So what happened over many years are a few things really done by many people, but where those 2 papers and what we did and what I tried to do subsequently would make a difference. The blood vessel inhibitors. So I told you our paper was 1976. It took 28 years from that paper before the first angiogenesis inhibitor would get approved. But starting in 2004, something remarkable happened. Avastin gets approved. Avastin is the number 2 or 3 best-selling biotech drug in history. And one after the next blood vessel inhibitors are approved. Even last year, it was the 4th biggest selling drug in the world. And not only are these drugs widely used for cancer, Nature, the journal estimates 500 million people will be treated with these. These are really the only treatments, pharmacologic treatments, for diseases of blindness in the back of the eye, like macular degeneration or diabetic retinopathy. These are drugs like Eylea and Lucentis. The drug delivery work, well, there I thought, gee, we'd done this work. I was so naive, I thought people would build on it. But really, I got large companies involved for a little bit, and they kind of gave up. So I got the idea of starting companies myself. I kind of learned if you're not your own champion, nobody else will be. So we started companies over the years. That also led to many different products. Probably the best known of them is this one, which some of you may have had. That's the COVID vaccine from Moderna, which I was able to start with Nubar Fayan, Ken Chen, and Derek Rossi. But there are many, many others there that we've helped on, and like everything from drug-eluting stents to new treatments for cancer, schizophrenia, and many others. And then the one other area that we started, and again, all my work is at the interface of kind of materials and medicine, is that you could someday, maybe you could make organs or tissues from scratch. And that would lead, on this last slide, to everything from new skin for burn victims, actually blood vessels, which Laura Nicholson, one of my students, started a company on, Humacyte, and that actually got approved last December, and they're actually used quite widely now in the Ukraine for treating wounded soldiers. And but not only can you do these for all these tissues, we're also working on organs and tissues on a chip. And of course, the most recent one and the beauty, I should say, of organs and tissues on a chip is if you try to do human testing, it doesn't go real fast. It's hard to control everything. If you try to do animals, well, animals aren't people. And so, but if you could do it, really make authentic systems on a chip, that might give you an opportunity to find new drug targets and so forth. And working with Li Wei Sai, this wonderful postdoc, Alice Stanton, and who's now a professor at Mass General and Harvard, who's actually here, this just came out a couple of days ago, she's actually made a brain on a chip where you can actually, and these are personalized brains. She can take any of your cells, convert them into what are called iPS cells, and convert them into the 6 different brain cell types. And she found a matrix, so they'll reorganize, and they make mini brains. At any rate, we're working on a lot of these, and that's what we try to do. We're also doing a lot of work with the Gates Foundation to try to bring all these new technologies to the developing world, including better nutrition and so forth. So I'll maybe stop here and see if I can answer a question or two before we go to the next talk. But I'm really honored to be able to present something, in the honor of somebody who's done the remarkable things that Saul Hertz has done. Anyhow, any questions? Okay, well then maybe we'll continue. So then the next presenter is Ilya Gip, who's at GE Healthcare, medical officer for oncology, and he's going to talk beyond the half-life, linking a discovery of the past to the future of cancer care. Sure.
If I can get my slides, and thank you for the sound. Yeah. Now, I do represent GE Healthcare. I'm the chief medical officer at the company, but I'm not going to be talking anything commercial. So this is not a commercial event, and I really try to get the title really beyond the half-life and linking the discovery of the past to the future of cancer care. Now, where I would like to start is the breakthrough innovation that took place 84 years ago. There is a picture right here. I think everyone in this room is familiar with what this picture is about. But I think that there is more than just the radioactive iodine. And if I were to summarize on what I really consider important regarding the real breakthrough, in my view, is number one, this, to my knowledge, was the first targeted approach in medicine. Now, if I go right now and Google targeted cancer care, the first thing that pops up is 1970s and tamoxifen and breast cancer. But really, the targeted approach, because of the ability of iodine to reach thyroid, was really pioneered by Dr. Saul Hertz. Number two is this was really one of the first approaches toward what we call today precision medicine. Realization, assessment, and response prediction, because that device that is also shown here and that is being touched by Dr. Saul Hertz is something that enabled clinicians not only to inject and administer, but also to see how the distribution took place and if this really has the therapeutic effect. Number three is that this was indeed the first radionuclide therapeutic use. The first radiotheranostics concept, because, well, maybe at that time it was not really necessary and there was no equipment that allowed people to really visualize. There was no SPECT CTs. But through iodine 123 and iodine 131, this can really be considered the pair that can be used for diagnostic and for therapeutic applications. And even used today, maybe in some challenging cases for thyroid cancer, but definitely going into MIBG. And the last but not least is it laid the foundation for personalization and adaptive therapy strategies, again, with the fact that Geiger counters was used to assess and measure the effects and the distribution of the radioisotope after it was administered. So going on further, it really took off, but it took off in an interesting manner, because the first publications that came out actually from Montefiore in New York, Dr. Sadlin, he consulted with Dr. Saul Hertz, and he understood that this is where it can really give a further advancement into cancer care. And indeed so just a few years later, you can see that things started to be quantified, things started to be objective, and started to be actionable so it could really be populated throughout the medical community. Now, this is something I wanted to brag about, and this is actually a part of what I believe was a beautiful coincidence. Because at that time, the more known isotope was iodine 128. Right, discovered by Enrico Fermi quite a few years earlier. Well, the point is that at that time it could have been used because it also has a therapeutic potential through the beta decay. But the problem was that it is having a much shorter half-life. What I did today in the morning is I did the run, and I did the run from the original building where the cyclotron was when the iodine 131 was generated to the Mass General building, or right next to that building, where it was administered. So I called this the first activity run. But the point is, I am not nearly as good of a runner. If this was iodine 128, none of the activity would have survived by the time that I would have gotten to Mass General. So the fact that it was practical, the fact that it was measured, it really laid the foundation for something that we're using today. And it's being used quite intensively. Now, I looked for some of the papers that are available today, and you can see that approximately 10,000 people of all ages are treated with radioiodine each year in the UK alone. That's their NHS data. So we're talking about hundreds of thousands and millions of patients being treated today. So something that started 84 years ago is with us today. There is also a lot of anticipation regarding radioactive iodine ablation therapy market forecast to continuously grow by approximately 5%, which I think we all would agree is not bad. And the most important is just recently, I think 2 or 3 years ago, the review paper came out on the strategies for radioiodine treatment, and it talked about the fact that radioiodine treatment represents the most widespread and effective therapy for differentiated thyroid cancer. Quite impressive for the targeted therapy that pioneered in 1941. So I think the legacy of this meeting is really talking about something that is so important that it has the future. Now, as of today, and I already mentioned the diagnostic and therapeutic pairs, obviously, as an example, a PSMA targeting agent, the linker and the payload, which is an isotope that can be used for either imaging, like, for example, the gallium-68 or the fluorine, and then linking this to lutetium-177, we can have the beautiful understanding of how the radiopharmaceutical is going to be affecting patients so that we can diagnose and treat with the same molecules, which opens the door for what we call radiotheranostics or radiotheragnostics. Now, I think that all are familiar with this image, and especially in the world of nuclear medicine, this is something that is very obvious and very well known. So each 2 images are representing a patient, the metastatic castrate-resistant prostate cancer, and then the second image is showing the patient after PSMA lutetium-177 treatment. Now, I will not talk just about this image because I think that would be unfair, and that would also hide away some of the challenges that lie ahead of us. Well, number one is if we're looking at something that happens today, not all of the patients are those beautiful super responders. Now, this is something that is the paper that also relatively recent that talks about up to 50% of patients who receive lutetium PSMA do not experience a PSA response. There is something that is to be done. Now, that's on the clinical side because we don't want to waste time, we don't want to waste money, but it is also on the economical and financial side because what also heads our way in the cancer care space is what is called OBA. Outcome-based agreements and outcome-based arrangements when the payment is being done not on the fact of the treatment that is being performed, but on the fact of the patient responding to the treatment and the clinical efficacy and efficiency reached. So there is multiple countries, multiple places on the planet where this is becoming a reality. It is not yet that strictly connected to radioligand therapies. Nevertheless, for expensive therapies, right, like radioligands are, it is definitely something that is to be taken care of. And if there is no prediction on the response, if there is no guaranteed outcomes, I think this also may potentially slow down the adoption, especially for the newer isotopes and newer radioligands. The next thing is the necessity for individualization, because it's not just about predicting the response. As it happens today, very often it's just the same dose, the same activity that is being injected in different patients. Well, I don't think I have to prove anything by showing patient 1 and patient 2 that those are different patients. And we can talk about the weight, we can talk about the blood volume, we can talk about efficient blood half-life, but the point is that if the patients are not the same, then how can the dose be the same, and how can the activity be the same? Now again, dose and the activity is not the same thing in radioligands, and Dr. Saul Hertz understood this when he was making the measurements and the notes that he was taking that Barbara already shown. Now, the question is, the personalization of radioligand therapy is naturally also coming with the fact that even the cancer itself could have a different volume, could have a different amount of cells that are cancerous cells, but it's not just the volume itself, which can relatively easily be measured by today's imaging technologies, but it is also the homogeneity or the characteristics of the tumor that is also affecting the way how the tumors are going to respond to the treatment. The other thing, and the last one I'm going to show in the necessities and the challenges that are laying ahead of us, at least on the clinical side, is actually here. So what you can see is those are the 4 bars, so for the lesion. So the first response from the first injection administration is very high, and then it's lower, and then the third and the fourth. And if we're doing 6 treatments with PSMA lutetium, well, quite obviously, so the efficiency goes down. So whether it's needed to really repeat it or maybe send patient for a short break or a longer break and still manage the way how the patient is living their life and the quality life, I think this is all ultimately what is going to happen in the field of radiotheranostics. Now, the other part is the complexity, which by itself is from the clinical side comes the complexity, but also on the physics of the radioisotopes. Now, I mentioned about radioligands, and this is where Saul Hertz started. So iodine 131. Now, I'm only talking about the therapy isotopes, so for example, Y90, maybe 223 radium, and of course, nowadays it's 177 lutetium. I am not familiar of many more, you probably know more, but you can see that out of all isotopes that are known today, and each block represents an isotope, we're just scratching the surface. There is no evidence that many other isotopes will not play the role. So moving on next, there is also a growing number of combinations in radioligand therapies. Maybe alpha particles, maybe beta particles, and maybe different molecules, small molecules. I just had a discussion prior to this meeting on the smaller ones, but also antibodies for immunotherapy and immunoresponse. Multiple radioconjugates, and then also multiple sequences, whether that's going to be a single injection, sequential courses, various cocktails, and the ability to move forward, but also on multiple synergies, like on the combination therapy, because today cancer care is no longer a monotherapy itself, so therefore we see that potentially with chemotherapy, immunotherapy, definitely with radiation therapy, especially for indications that are well known today, we're coming to thousands of different combinations that will require attention, and this complexity cannot be solved by a single entity. That's why partnerships are so important, and that's why I'm so appreciative to be a part of this meeting. A part of this is also consortiums. Now, this is one of the consortiums that I have joined as a company and also being a part of the Oncinium Foundation. Thera4Care Consortium is looking at the diagnostic innovation, therapeutic precision, data-driven insights, and elevating the knowledge and raising questions that potentially could be solved, whether the IT, AI technologies, imaging innovation, innovation on the clinical sides. So there is more. This is a European initiative, but I don't see the reason why being on this side of the pond is not also bringing us to the same conclusions that this is necessary. And promoting dialogues is very important, especially when we speak about synergizing the potential of various therapies. We're looking about the possibility, and this is the paper that just came out. I'm very proud that I co-authored this paper, defining synergy of integrating radiotherapy and radioligand therapy. And this is the outcomes of ESTRO 2025. And please look at this. This is the symposium that we ran. At the beginning of the symposium, I've asked the question to the audience, asking, how prepared do you feel to integrate radioligand therapy alongside radiation therapy? And the upper bar is actually showing the answers before the discussion, and you can see that the majority is really saying not ready or no formal plans. And then after the discussion, just by having a simple dialogue, sharing the knowledge, giving the talks, it changes dramatically. Therefore, the importance of the dialogue is so necessary. So linking a discovery of the past to the future of cancer care, I believe something that started in 1941 is shining today, and what I call radiating hope for the future of cancer care. So the legacy of Dr. Saul Hertz, we've got a lot of other things to do, and I'm so appreciative of Barbara to inviting me here. Thank you very much for your attention. And I would like to introduce the next speaker. Next speaker is David Gilmore. He was a president of SNMMI, the Massachusetts College of Pharmacy and Health Sciences. He is the associate professor of nuclear medicine, and he will give a talk on theranostics, building a critical workforce. David.
All right, hello everyone, welcome. I will agree with everyone else, except I did not do the run this morning from MIT to Mass General. Not on my thing. But what I want to talk about today is you're going to continue to hear the great excitements and what's happening around theranostics, where we start, where are we going. But we also got to really think about how we're going to do this from a workforce standpoint. So the first ever theranostics kind of summit was put together by the Society of Nuclear Medicine and Molecular Imaging. Several people in the room was actually at this and in attendance for it. This here, the part that I'm in, is one group that was really kind of focused on workforce development and where and how we're going to be able to administer this to patients. There is a lot of things that are challenges that have been recognized right now. First and foremost is starting kind of at the forefront of the patient, the individuals that is actually administering the radiopharmaceuticals as well as doing the imaging is nowhere near prepared and ready to do this. And at the same time, after COVID was a massive kind of hole that was left in the actual workforce itself. At the same time, and you can pick up the news at any point today, seeing that colleges and universities are under extreme pressure to reduce cost. And so traditionally, when we've had small programs at colleges and universities that are not as well endowed as some others, those programs seem to go by the wayside. And so I'm going to show you a little bit of what the challenge for that is. But the barrier is, how do we get everyone ready for this? You would think over 84 years that we've had the opportunity to figure out how to administer radiopharmaceuticals from a therapeutic standpoint, and we did. We succeeded really well with that. But you just heard, there's a lot of new agents kind of on the market that's really kind of expanding what we had to do with I-131 and where we're going. So I'm also on the board of directors for the Society of Nuclear Medicine and Molecular Imaging, and one of the focus that I am kind of leading at the moment is really around the workforce development. And so this is some survey data that really went out to physicians, technologists, scientists, and radiopharmacists, really looking at where do you see we're at and what we're doing. So the first one here, sorry, let me turn around here. The first one is looking at who is going to be leaving the market within the next couple of years, 5 years and 10 years. And you can kind of really see, particularly from the technologists in the science section, we have more than half of the workforce preparing to leave the workforce. The flip to this is, well, what do we currently have in place? And what this is showing is that many of our nuclear medicine programs, from an applicant standpoint, have somewhere between 11 to 20 applicants per year, and most take somewhere between 5 to 10 students. Now, as a reminder, half of the current workforce is planning to leave in the next 1 to 5 years, so this is not going to meet that demand. The next is we have the accredited programs kind of across the country. Every state that is kind of dark here have no programs at all. The states, oops, do I go backwards? Let's see. The states that are kind of in this greenish color, for the most part, have one program in each state. Again, somewhere between 5 to 10 students. This you can just show from actual data of who's taking the certification boards has drastically reduced, even from 2012 to we have less than somewhere around 60 to 70 kind of with that. We are increasing a tad bit, but not a lot with it. This map shows currently where we are with programs. That's the blue dots. The red programs may mean that we have students doing distance learning, but you can see there's a huge part of the population that we do not have the resources to do that currently. This is looking at, again, from that data that showed how many of you are leaving the workforce and where the holes are going to be, is what do you think the perception of this is as more and more radiopharmaceuticals are hitting the market for therapy? And what we see with this is that, the majority believe in any category, whether you're a scientist, radiopharmacist, technologist, or physician, believes that there will continue to be an increase in this. That also means that the current stuff is not going away, but will continue to be added. Again, highlighting the fact that we're not prepared from a workforce. This is now looking at that same data went out to the actual hospitals and imaging centers and oncology centers, looking at, okay, so where do you believe you're going to be hiring and where is this coming from? Again, you can kind of see, particularly from the physician and the scientist section, huge numbers of people wanting to increase the number of individuals from a workforce. But again, those programs are not there to support that. So really what the Society of Nuclear Medicine and Molecular Imaging from the Theranostic Summit really focused on is we need a plan, right? This can no longer be, yeah, it's coming in the future. No, it's here now. And you just heard how many approvals that we've received in the last couple of years. You're going to continue to hear that throughout this. So year one is really developing kind of where is that model going to be? Where are we going to be able to educate, train these individuals? How are we identifying the grassroots to do this? Whereas by group year 3, we really need to be focused on this from a global standpoint. It is not just a US problem. This is something that we see with our colleagues and sister societies and other organizations as well across the world. And then kind of by year 5, it's time to reassess this as we know this development and the FDA approvals are going to continue to hit more and more. No worries. The other thing that we have is access and the challenges that we have to that. And so what we see here is just kind of where we're at with the centers actually able to do this. And finally is that if we're going to continue to expand our plan and get more patients having access to this, we got to really think through who is the team doing it, how are we going to kind of get to that point. So it's just showing you there's a ton of committees really focused. There's new initiatives kind of going out to really do this. And finally, just to kind of as a FYI for everyone who's interested in theranostics, is the summit coming up to continue to learn how we're going to do that. I unfortunately can't be a part of the panel this afternoon, or I don't think I'm going to, because I'm doing another theranostics talk. But if you do have questions, I'm happy to answer. Just please fill in, email me that. So thank you very much. And my great pleasure next is to introduce Anika, who I've had the great privilege to not work directly with, but side to side as we're across the street from one another at Dana-Farber Cancer Institute. So, Anika. Thank you. Oh. So this moves the slides and points.
Okay. Yeah. Can you hear me? Yeah? Okay, great. Well, thank you very much for the invitation, Barbara. I cannot thank you enough. It's always a pleasure to see you, but it's really a great honor to continuously honor your father. I think that he really left a legacy that we are all benefiting from. Sorry, I thought I was wrong. No. These are my conflicts of interest. The only one relevant to this talk is my relationship with Imaginab, where I am a site and imaging PI on 2 phase 2 trials, a part of the SAB, and a consultant. So I find it rather, somehow the slide didn't come true, but if you actually look, I don't know if you can fix that. Can you see the slide on your? The slide is not coming true. Anyways, it's a timeline, a kind of history, bringing the time back to 1941 and illustrating that there's been quite a gap, and this is looking at PubMed data for theranostic or theragnostic. And it really is only in 2000 when the field of precision imaging, precision medicine, with the introduction of molecularly targeted therapies, all the omics, choose your favorite, and theranostic came on board. And then with immunotherapy 10 years later, those modalities, which are now called pillars of cancer treatment, have been validated by multiple publications. But I think one thing to emphasize is also the importance of the 3 publications that Dr. Hanahan and Weinberg brought in 2011 and 2022, because they are really visionary manuscripts, and we are living in what they predicted right now. So the names, as you may know, were cornered by John Funkhauser, who was the CEO of Pharmanetics in 1998. And it combines diagnostic methods with personalized therapeutic approaches that has benefited since the early concept of targeted drug delivery. There is an issue with the projection, because there should be a picture on the right. Can you, I don't know if you can fix that. It seems that none. Can you press a button again? Says Dr. Anyways, what that picture was showing, okay, that picture is not showing, that is the field is actually 3 major fields. You know, the theranostics can use imaging methodology, but then it delivers the load either with radioactive compounds, with nanomedicine, or with cellular components. And if I need to define radiotheranostics very simply, if you can see it, you can treat it. And you would have seen Dr. Saul agreeing with me right there if the slides were working. I hope this is not going to, I tested it before I came, and so I hope that some of the images are going to come true. Otherwise, I'll ask somebody else to maybe step in for me now and then try to fix it, because that's not going to help. So I don't know if Dr. Price is here. Oh, there she is. I hadn't seen you. Dr. Price, would you mind stepping in for me and see if I can fix that? Hold on 1 second. I'd like to introduce you, and I have it on my phone. So it is my great pleasure to introduce Dr. Price, who is a colleague of mine in the sense that she's also at Harvard Medical School. But Dr. Price began her academic career at the University of Pittsburgh, where she raised to the level of professors, professor of radiology and biostatistics and the head of PET methodology. She then joined the Mass General in 2017 and became professor of radiology there. And her research, and she's very well known for that, focuses on developing and applying PET techniques to quantify ligand protein binding, neuroreceptor systems, metabolism, and cerebral blood flow in the living brain. She has been instrumental in advancing these imaging methods, and especially in neurodegenerative disease such as Alzheimer's. She played a key role in the development and validation of a compound called the PIB, PET imaging compound in Alzheimer's disease. And her contributions have been recognized widely across the field. She was named a Fellow of the Society of Nuclear Medicine and Molecular Imaging, and in 2025 received the Society's prestigious Paul Abersold Award for Standing Achievement in Basic Nuclear Medicine Science. I can go on and on, Dr. Price, but, but thank you for being here.
Okay. No, I don't think I need it. Can you hear me? Can everyone? Okay. So really what I'm going to do in a short period of time is tell a story about my experience with nuclear medicine, and I hope that it reflects the gratitude I have for having the opportunity not only to be here today, but to participate in this research. Okay. So nuclear medicine and molecular imaging, what I'm going, okay. Is this better? Okay. No. Okay. So I'm really going to focus today on positron emission tomography and just talk about how we leverage the positron emission process, working with a group of very talented people that enables us to study normal and pathological processes at the cellular and molecular levels with very high sensitivity. And we benefit because several radionuclides, as listed here, are positron emitters, and that facilitates incorporation to amino acids, carbohydrates, components of molecules and drugs, and allows us to use them to trace biological processes. So there are just many things that we study, and I've had the great fortune of being able to utilize PET for many of these purposes because my background mostly is technical. And this is just an example of some of the radio tracers and the specificity that we're able to achieve when applying PET to target proteins in vivo in the living brain. And there are many applications, as you know, for neuroimaging with PET. And today I'm going to really focus on neurodegeneration. Two examples here being the beta amyloid radio tracer, which is one of the hallmarks of Alzheimer's disease, but also a very important neuroreceptor system that has been imaged in Parkinson's disease, the dopamine system. And what I'm going to move on now is just to talk a little bit about small molecule PET tracers for proteinopathies. Under normal physiologic conditions, there's homeostatic equilibrium, but there's just a variety of diseases that really can perturb this equilibrium and impair cellular functions. In particular, is amyloid beta, tau, and alpha-synuclein. Today, this being a Venn diagram of 3 aggregated amyloid proteins in neurodegenerative diseases, and the overlap is what also brings a challenge, and the radiochemistry colleagues are really charged with developing selective radio tracers. So the hallmarks I'll be speaking about in terms of PET imaging are amyloid beta fragments, 1 to 40 and 1 to 42 amino acids long, as well as paired helical filaments or neurofibrillary tangles that contain the tau that becomes it's intracellular, microtubule. With hyperphosphorylation, they can become aggregated. The amyloid beta is generally extracellular, and that results from the cleavage of amyloid precursor protein with the gamma beta secretases. And this is just the neuropathological staging that when these ligands were developed, that we really followed in terms of the knowledge that we had in the field at the time, showing amyloid deposits initially appearing in basal portions of isocortex and then distributing at later stages to all areas of isocortex. And Brock and Brock also showed that neurofibrillar changes, or tau-containing tangles, began in this work. They identified transinterrhinal origin and then distributing to isocortex. So different patterns. And it's amazing to me that it's been almost 20 years since the original highly selective imaging of amyloid in control and AD individuals. On the right, the Alzheimer's disease subject shows the typical FDG PET or hypometabolism that was the standard in the field at the time for Alzheimer's disease. And in that same individual, the amyloid PIB scan clearly shows nearly twofold greater uptake in areas of cortex relative to the nonspecific uptake in the control, who exhibited normal or healthy glucose. And what in this field, I think for the last 20 years, I think we've really been focused on how early can imaging detect the pathology. And that's really driven the field. We know what the major risk factors are. We know it's age, the risk for a female or male at 45, maybe 10 and 20%. Females have higher risk. But after the age of 80 or 85, that risk really, really increases. And we know that individuals who have ApoE4 allele are more prone to the disease. So we've really focused on moving this imaging field earlier and earlier. And this is just examples on the top of very early amyloid, the spectrum that we observe in normal. It's normal controls, but I don't know that that really has a meaning. So, but cognitively unimpaired individuals on the left, the mild cognitive impairment, individuals who've been diagnosed with cognitive impairment that can be prodromal to Alzheimer's disease. And you can see how an individual with mild cognitive impairment can harbor maybe no amyloid or have as much as someone with Alzheimer's disease. And that's really what we've been focusing on, is to understand what the tipping point is. And about 2013, tau PET became available, and this is also a representation of the spectrum of uptake in living individuals, starting from the left for someone who is thinking normally to someone on the right, in G, who has an extraordinary amount of tau deposition in the brain and very impaired cognition. So over the 20 years, there's now 3 fluorine-18 labeled FDA agents, amyloid imaging agents approved for human use. This has really dramatically helped the field to grow. And there's only one FDA-approved tau radio tracer at this time. So there's more developments to come. Although the radio tracer fluorine-18 MK6240 has very strong characteristics, and it's being more widely used. Just moving along, I'd be remiss if I didn't say something about the Alzheimer's Disease Neuroimaging Initiative, because I think it's really impacted this field, the progression of the field, and it's been in existence over 20 years, and it's an extraordinary, successful collaboration with academia, industry, private sector, and more than 6,000 publications have resulted from this study. And it's really impacted the field in terms of developing standardized biomarkers, standardized protocols, inspired initiatives in many different areas, and it acted as a data sharing model that was very important in this field, as well as the collaborations and the research productivity. And it's also inspired partnerships for Alzheimer's, for other neurodegenerative disorders, including Parkinson's disease and multiple sclerosis. And many of you have seen this model, but this this results directly, in my mind, from ADNI. The Mayo Clinic researchers developed it and published it, but it just really puts in a graphical form the sequence of events that are so devastating in Alzheimer's disease, beginning with the accumulation of amyloid that can be detected with cerebrospinal fluid sampling or PET, then synaptic dysfunction, FDG PET or fMRI, magnetic resonance imaging, and then tau-mediated neuronal injury follows, and much later is brain structure, atrophy, cognition loss, and loss of clinical function. So it's not until the very end that the symptoms are apparent. And there have been efforts over years to upgrade or update the criteria for diagnosis that really moved it from clinical symptoms to biology. And the question being, how does amyloid relate to tau? What is that tipping point? The thought is that it's the accumulation of amyloid is there. Amyloid is first. Tau can be part of normal aging, but what is it about that tau that propagates? When it seems to propagate into neocortex, that may represent part of that tipping point. And that's where we are now. And now we're working on staging criteria and validating those criteria in vivo. And just to put things in perspective, it was 1951, 1971 for radioactive iodine therapy, hypothyroidism, and then for therapeutic cancer treatment. And I remember when I first was part of this field, I would calibrate the thyroid machines, the instruments for thyroid assessments when I was at George Washington. So in terms of what's been FDA approved in brain, it's really been mostly stroke, and it's not until just recently that the amyloid radio tracers and the one tau tracer have been approved. So just ending with a look at the future, the prevalence of dementia is staggering, and this suggests that it will be 3 times higher when based on biological rather than the clinical symptoms. So we really need to evaluate earlier, move from today where the diagnoses have been clinical symptoms to the future, where they're biological constructs. And we've seen the fruit of those efforts with the approval of 2 anti-amyloid therapeutics that are available for use. And finally now, it appears that a PET radiotracer for alpha-synuclein is going to be realized. And I know that there are efforts for TDP-43 relevant for ALS that are emerging. So the paradigm shift has been important over 20 years, 25 years that I've been seeing from neurodegenerative, within neurodegenerative diseases. And just to summarize, this project has really been remarkable in terms of nuclear medicine and neurodegeneration, and it really has represented a strong effort by many entities to get us to the AD therapeutic stage. And we're really looking forward to the next phases with alpha-synuclein and TDP-43, so that today is yesterday and the future is today. Thank you. So I am a bit embarrassed because I don't have the, yeah, that's what I need. Thank you. Oh. I did, but I don't have the detail. I definitely don't have the detail. So. So. No, I apologize for this. So the next speaker is Dr. Jason Deerling, head of radiopharmacology at Mariana Oncology in Novartis, and he'll be speaking on targeting radioligand therapy for the treatment of small cell lung cancer and beyond.
Thank you. Thank you, Professor Price. That's very kind. Yes, I'm Jason Deerling. I'll be talking to you this afternoon about our work in discovering and developing a DLL3 targeting RLT for the treatment of SCLC and beyond. So I do have some disclosures. The most important one is top left, as an employee of Mariana Oncology, as you all know now, which is a Novartis company since the acquisition last year. And at Mariana Oncology, we are, of course, very well aware of our debt to the legacy of Dr. Hertz. Soon after we moved from our Soviet chic initial laboratories out in Woburn, we moved to Watertown, which some of you might know, just behind the Home Depot. Just after we moved there, Barbara very kindly came over, and we were delighted to have her open our Saul Hertz seminar room. So that's our portrait and the plaque. We've all seen this portrait several times this afternoon. And that's his view. So we're overlooking not just the parade of trees along the Charles River, but also Harvard and MGH. So he's overlooking his old stamping ground, which is rather nice. And that's where he started this subject of which we would now call theranostics, which is summarized here. Not much new information here for this crowd. This is an RLT. We can see the radionuclide at the top is being delivered to the tumor by the linker and chelator and the ligand, which is binding to the tumor cells. So really, this slide is just to establish that we are at Mariana not an alpha or a beta company or a small molecule or an antibody. All we do is we want to target the tumor, deliver the cytotoxic dose using the best PK, the best vehicle that we can. And this is how we do it. I'm not going to go into too much detail on this slide, but this is just to represent all the different functions of the company. We go all the way from discovery on the left, through development in the center, and then out to delivery. So in-house, we have all these different functions. Biology, is this going to be a good clinical unmet need for us to address? Can we do so? And then discovery biology, how do we identify the best vectors? And then radiopharmacology, making sure that we've got the optimal PK, we can achieve that therapeutic index. And then through to robust manufacturing and supply, and we've been sending out our own clinical doses over the last couple of weeks. So we've truly, over 4 years, we've managed to fulfill all these criteria. So why DLL3? Well, delta-like ligand 3, it's emerging as a very attractive target for small cell lung cancer, a relatively small portion of the lung cancer patients, but it's, as it says here, a very aggressive cancer. Extensive stage is really a very aggressive cancer. It has a 5-year survival rate of about 9%. But we do have this opportunity. Now that we've identified a clinical unmet need, DLL3 is expressed by 80 to 90% of small cell lung cancers by immunohistochemistry, and SCLC is highly radiosensitive. So we have a gateway and an opportunity. This graph also shows a couple of carcinoid and neuroendocrine tumors. There's a lot more heterogeneity between these patients, so we're going to need a way of identifying those, but we know not only can we address this clinical unmet need, we can also extend our findings and benefit other populations of patients. So how do we do that? I've alluded to this already, the discovery and the development portion. Here we're going to be talking about how we worked our way towards designing this macrocyclic peptide. So we have this in-house mRNA display. They use all sorts of tricks to speed up this process as well. And then we get to the point where we have some hits. So we'll have various different series. We'll pick good ones for different reasons. We have a target critical pathway. And then we end up with things like compound A. So here we have a macrocyclic peptide, which we know does bind somewhat to our target. It's 12 nanomolar, so I'm going to be throwing out some numbers. The way to think about this is a micromolar binder will be binding to the surface, not much opportunity for improvement there. And then as we improve the affinity, it's finding a cleft and it's starting to bind there. And then we're really refining how it's binding to those various different residues in the cleft on the protein. And that's where we get down to the picomolar point. And this is the process, part of the target critical profile that I was talking about. We're going to improve the binding affinity by changing various different residues. We're going to ensure the proteolytic stability. We're going to be delivering this systemically. If we inject it into the blood, it's got to have a reasonable half-life of stability. And then we're going to be identifying the site for the DOTA attachment. And all of these things can happen in parallel to some degree. Once we've got some nice binding in vitro and we're demonstrating cell binding and internalization, we'll then think about doing things like biodistribution studies. And we can see here a percentage of injected activity per gram. So we're getting how much radioactivity is in which of these tissues. And this is another step on our pathway. Still not the final product, but we've got at 24 hours, which is a nice time for us to think about a therapeutic delivering its dose to the tumor. We've got very little in the blood, so we're not going to be having an issue with bone marrow toxicity, for example. Liver is clearing nicely. It's a peptide. Clearly, we're going to have some kidney uptake, but we're going to be addressing that later on. And then we've got some uptake in the tumor. And this is real uptake. When we're down at 0.1%, people start to get excited, but at this sort of level, we now know that we're heading towards an inflection point. Importantly, we also have some uptake in the tumor at 72 hours. These therapeutic nuclides that we're mentioning, lutetium and actinium, they've got half-lives of about 6 and 10 days. So we want to keep them in the tumor for longer so they can deliver more of that energy and kill more of the tumor cells. And this is just a bit of a window into one of those steps. By solving the co-crystal structure, we know where compound B is binding to that particular target. We can then say, okay, do we lengthen this aliphatic chain so that we can improve this interaction? Do we shorten it so it's not interfering with this? Where should we put the DOTA? Are there any waters that we need to pop out the way? How can we best improve that K-on rate so that we're binding, improve the K-off rate so that it's not coming off, and then that inflection point is really where it's staying on the tumor cell surface long enough that we're going to get some internalization, and that's where we really improve the area under the curve. That's where we deliver the most grays, the most toxicity to the tumor. So that's all great, and we got to ETN-029, which is our final molecule that we developed as a process. All this, it took us about 1.5 years of development from deciding on the target to actually arriving at this. And this is just summarizing many studies, but you can see it's a very stepwise. There are a few missteps, and then we find out that that wasn't the right way to go. As we improve the tumor to kidney ratio, that therapeutic index that I was mentioning, and the tumor uptake at 24 hours as a nice snapshot of whether this will be a useful therapy. And we got to compound F, which was great. It's a biotech company, and we now had a product. So this was our first developmental candidate. We had a nice tumor to kidney ratio of 2 to 1. Everyone was very happy, and we triggered all sorts of other things, which are then long-term, very expensive commitments. But we're looking at, reasonable amounts of money and a 6-month commitment to develop compound F and make sure that it's going to be non-toxic in higher species and all these sorts of things that we needed to do outhouse. As a biotech company, and we already had these various steps in place, we had some backup molecules. Because if you have all this machinery in place and then you find out you have an immunogenicity problem, you can just change minor parts of the molecule and still progress to a clinical stage. But ETN-029 was, as you can see, significantly better than F. And this was a very, very small change that just improved the renal clearance by about 5-fold. This was a tense discussion I had with our CSO when we got this 24-hour data, and he said, no, that causes me all sorts of problems, go and do it again. I didn't want to do it again, so I did the 4 and the 72-hour biodistribution data, and sadly for me, they were in agreement with this. This is what ended up in the clinic. So we went to the leadership of the company and also the board, and we said, this is the position. Yes, we are committed here for this molecule. We'll learn a lot from it. This is actually the better molecule. And this is typical of Mariana Oncology. You will hear the word patients in the company a lot. It's all about the patients. And the board said, yep, absolutely, that's the right thing to do. As you can imagine, and some of you have slightly more experience of this, in a biotech company, to say we're going to take a 6-month hit was not insignificant, but it was the right thing to do. So we ended up with ETN-029, which, as I say, is heading towards clinical application. Very exciting. I mentioned we collected other time points for the biodistributions, and this is just showing the sort of biodistribution that as a radiopharmaceutical development or in any other drug, this is what you love to see. At 2 hours, very little in the blood and lung, and then clearing from all the normal tissues. Some in the kidney, clearing very nicely, but very good retention in the tumor. So now we can deliver that dose to the tumor. Very exciting. It's in the tumor. Where is it in the tumor? Because we know we need to get that really nice distribution throughout all the tumor so that we're hitting all the tumor cells and we don't get any regressions. These are autoradiography images of lutetium delivered using ETN-029 on the left, and you can just see there's a nice homogeneous spread throughout the SCLC xenograft tumor. For comparison, this is a DLL3 binding antibody on the right with a very heterogeneous distribution. This isn't going to get to all the tumor. There's going to be some areas that are overtreated, some that are undertreated. There's going to be repeats required. So this is showing that with our peptides, we're getting to all those viable tumor cells. And what does it do once it gets there? On the left, we can see that the control tumors continue to grow after treatment on day 12, but with treatments of 0.25, which was less successful, and then 1 microcurie of actinium-labeled peptide, we were able to control tumor growth in a dose-dependent manner. Very nice. So that's SHP77. I do want to draw attention to these SHP cells are expressing this target at 3,000 copies per cell. The targets we would normally approach are expressing at more in the range of around 100,000 with some hand waving copies per cell. So at 3,000, this would be considered negative for that target. And then on the right, we can see in the H69 xenografts, these are expressing at approximately 1,000 copies per cell. So now we're really pushing, okay, how many of these patients can we help? And this is showing that we're getting a response, we're getting tumor growth control in that very challenging H69 target. And as I mentioned earlier, there are other patients that we would want to help, patients with carcinoid, neuroendocrine. Are they expressing this tumor? So for our SCLC patients, we can see with this indium-labeled peptide, nice uptake in the tumor on the right shoulder, very little activity remaining in the kidneys, clearing via the bladder at 6 out to 72 hours. So here we can see for our SCLC patients, we can not only confirm that they're expressing the target, we can do individual dosimetry, which has already been alluded to this afternoon, and then we can monitor the response over time. We can also identify within that very heterogeneous population of patients who, with carcinoid, with neuroendocrine, et cetera, we could also help. So in summary, it's been an exciting 4 years for Mariana through various different stages. Actinium ETN-029 is a first-in-class peptide-based RLT targeting DLL3 expressing tumors. We've been through how we work through this target critical profile, structure-based drug design, experiential, shifting around the DOTA. We've learned all sorts of things about how to target these, not just this target, but various other targets as well. We've got excellent tumor uptake, a nice favorable tumor to kidney ratio, and that a single dose of actinium-labeled peptide controlled these very aggressively growing tumors. And as I mentioned, we're now a clinical stage company. We're sending out from that building in Watertown that you visited, we're sending out our own doses, and you'll hear the gong. This is a lot of the gang. I'll just bring your attention to a couple of them. So here we have Alonso Ricardo. It was Alonso and Simon Reed. Alonso was our CSO, and Simon Reed was our CEO, and it was their plan. They started with a blank piece of paper and said, okay, what can we usefully do with peptides? And they started that in the beginning of 2021. So you can see how quickly that moved. Simon's parting gift to us was that he left us a gong that we sound every time we send out a patient dose. So that's what you'll be hearing as you drive along I-90. And thank you for your kind attention. Yep.
Okay, I think we have fixed it. I'm running it from my own computer rather than the MIT computer. Hopefully mine will work better. If we can switch, aha, we have a picture. The part of that picture was to really look at the literature on the subject that we're discussing today. And you can see that it took a while since Dr. Hertz started the field in 1941 until the advent of all those other pillars of treatment. Next slide. I can activate that. And I mentioned the significant, the origin of the name of the field, and what I wanted to show you was the differences in the theranostic approaches, having a radioactivity, nanoparticle, or cellular component contributing to that delivery. And all of that field has been benefiting from all we have learned during this gap up to 2000. So again, if you can see it, in my view, you can treat it, and I think Dr. Hertz would agree with that. And the field of precision medicine in which we live, this is again, PSMA has been mentioned before, but just as to illustrate that example, is to really get an imaging and a therapeutic agent that are so similar that the only thing you need to do is flip the isotope, because the chemistry remains stable and therefore the biodistribution remains stable. Using the imaging agent, you can derive an imaging that will actually demonstrate the extent of metastatic disease. All those little dots here are in the bone marrow, in the skeleton of that patient. This is normal distribution in the liver, the spleen, the kidneys, and the salivary glands. And then you can flip the imaging isotope that delivers precision imaging to the, in this case, the lutetium, the beta emitter, and derive a similar image that can be captured that shows a nice match of the uptake in following the therapy that matches the diagnostic image. And this is precision therapy. And so both of these provide essential information that we use in the clinic, but that are also used for drug development, confirming mainly the presence of the target so that you can find the patient eligible for that particular treatment. The other beauty of imaging is that you can look at the entire tumor burden at once, rather than just a single metastasis that would have been biopsied. You can look for heterogeneity, the biodistribution, the kinetics are important to evaluate because they also give you information about toxicity, for example. We can use this for staging, restaging, and all the clinical use that we use imaging for. But we can also derive dosimetry and then derive precise delivery of the amount of dose that the patient requires. So there's a lot of benefit from having that kind of approach. And this is the patient I just showed you that has the imaging study here. This is the image after the first dose of PSMA. You can see his PSA was pretty high at 387. And then the second dose is being given just a few months later, and you can see a marked decrease in the PSA that also is demonstrated by the imaging with only a few lesions left, and continuous improvement is seen with the second dose. Now you can choose beta, alpha, and Auger delivery of the load. They have different ranges and different power and punch. I have no time to go over these now, but there's quite a bit of choice there. And here is an example of a patient who was treated with lutetium. You can see that his PSA was even higher at above 1,000, significant decrease in the PSA. But then within a few months, the patient shows an increase again and reemergence of disease, then was enrolled in another trial that then tested actinium, an alpha emitter, as opposed to the beta emitter, and you can see amazing response relative to that treatment. So there is a way to actually look and pick and choose what would be beneficial to that patient. And that particular field and the current landscape is extraordinarily busy. This is a recent report of the number of targets being analyzed, and you see we went beyond neuroendocrine tumors and PSMA, which have led the work in that field to FAP, to other particular targets. The therapeutic radionuclides have moved from lutetium to yttrium, actinium, and so forth. Iodine is, of course, being used as well. The carriers have been polyvalent, small molecules, peptide, antibodies. You heard some of that earlier. And then we now have a wide range of solid tumors, including liquid tumors being tested with that approach. So, you cannot stop getting news on what it is. But I think it's important to come back to a principle that both Dr. Hanahan and Weinberg reported in as early as 2000, 25 years ago, when they published this paper, and recognized that the field had been very cancer cell-centric for decades. And also recognized that that focus had produced an extraordinary body of knowledge that we still benefit from today. But the field of cancer research, they say, has largely been guided by a reductionist, as they call it, focus on cancer cells and the genes within them. And they proposed in their paper to move from this reductionist view to what they call a heterotypic cell biology, where you can now look at endothelial cells that line that angiogenesis that you see in the tumor bed, and also to other cells within the tumor microenvironment, such as immune cells, fibroblasts, and other cells. But they also had a very interesting, I think, visionary comment in 2000 saying that the interactions between the genetically altered malignant cells and the supporting co-conspirators, I love that name, will prove critical to understanding cancer pathogenesis and to the development of novel effective therapies. And are we not living in that era right now? So the existence of the TME is a major reason underlying cancer occurrence, development, invasion, metastasis, creation of an immunosuppressive microenvironment that leads to drug resistance and recurrence after treatment. And again, interestingly, looking at the literature in that subject, when it comes to tumor microenvironment, it grows up the same way as theranostic has done after the advent of the landmark that I mentioned earlier. So I call it seeing the unseen in cancer because therapies targeting cancer cells specifically do become insufficient when the TME causes therapy resistance. And so there is a need to target both the cancer cell and the stromal component. And this may require a combination of technologies that may include more than one imaging modality or imaging tracers. Also the incorporation and combination with early phase technologies such as circulating tumor DNA. There's enormous data coming out there saying how early we can detect lack of response or recurrence, but where the heck is the tumor? So I think that if you need to act upon it, it would be nice to have an imaging agent that can help in artificial intelligence. And then as a practitioner in that field, I do feel that I do have a responsibility to provide a snapshot of what the whole cancer biology is to the clinicians in order to make the right decisions for that patient. A point that's quite interesting as opposed to the genes is the fact that the TME is also less genetically unstable. And so phenotypes can be shared between different tumor types and across several cancers. And this is something we are seeing with some component of the TME already. And so this leads to new avenues for designing novel diagnostic and therapeutics. We've been very busy addressing all the hallmarks that Dr. Hanahan and Weinberg have mentioned earlier, and can use them in many different ways. Some of them are FDA approved, some are all in investigational trials. But if you ask questions about immunity, stress environment, metabolism, or tumor environment, we can answer some of those questions already. But I think this brings up the issue of what is happening when we actually run a clinical trial, and how long does it take to come from point A, which is the drug discovery, to the end, which is access to patient care. And this is a slide actually that came, a graph that I found in 2008, but checking recently on what the timeline is, it turns out it has not changed. And the time between drug discovery to access to patients can be up to 15 years. Now, the fate of discovery and the pace at which we are living now when it comes to all the pillars of treatments, we went from millennia to centuries to 50 years to come from cytotoxic chemotherapy to molecularly targeted therapy, another 50 years to come to other advances. Now it's 10 years. It's much shorter. We do need to take that into account because we're wasting a lot of time, and those trials are lengthy. They are high risk with high attrition. The likelihood of approval recently for phase 1 drugs has not just been reported at 6.7%, and it requires a large number of patients. So I think that's a major barrier to the rapid translation of the scientific knowledge that we are getting now into clinical advances. And so we need to change the traditional benchmarking metrics, and they need to be strengthened. And so we need new strategies that can aid in the early selection of promising candidates to move to pivotal trials and terminate candidates that are unlikely to be successful. And we can provide evidence of biologic activity in the initial stage of the drug development and confirm on-target drug effects and identify candidates that are more likely to benefit patients. So we require for that to have repetitive, non-invasive, quantitative, longitudinal evaluation, the same way we would do it in a preclinical setting with mice. We can do that with humans as well. And I have the privilege, I have to say, by having been at the Faber for so long, to have been a witness of a first-in-man or first-in-woman novel drug that completely changed the field. I saw the first patient on imatinib. I saw the first patient on ipilimumab. I saw the first patient on this and that and that. I don't have time to go over that, but I just want to give you one or two snapshots of what we learned early on. Here's a patient with GIST, a gastrointestinal stromal tumor, who was receiving a drug at the time called STI-571. And you can see that that patient actually has extensive metastatic involvement through the liver, extended to the left upper quadrant, and you can see the abnormalities on the corresponding CT. Now, given the fact that we had seen the first patient within a month and saw a fantastic response with that, I decided to do it even earlier. And so 24 hours after a single dose of imatinib, we repeated the PET scan, and this is what we saw. And it's a completely normal FDG scan. There's absolutely no evidence of glycolytic activity within the liver. It's completely suppressed. And then 25 days later, the patient comes back and comes to us as being diagnosed with progressive disease, because traditionally the measurements are based on anatomic measurements on CT or MRI. And you can see that that patient actually has new lesions within the liver compared to baseline. So within a month, that patient is classified as a progressor by CT criteria. And I said, but wait a minute, let's do the PET. So we do the PET, and the PET is as cold, quote unquote, as we call it, as the 24-hour one. She then comes back 3.5 years later, and you can see that she still has quite a bit of abnormalities, but you can notice if you compare to the density of those masses in the hepatic parenchyma here, that they're becoming darker, which means that they're becoming more necrotic, and they're darker here, they're more liquefied. And yes, you see them very nicely against the normal hepatic parenchyma, but this is not a disease. The patient is still negative on FDG. She's still responding to imatinib. And so it's important to remember that metabolic response can precede anatomic response and be an indicator of the fact that your drug targeted actually hit the target and is predictive of longer-term response. And that same patient continued to come and visit us. She comes 5.5 years later. She still has an abnormal CT. She comes 10 years later, and she still has an abnormal CT. If you were to look at that CT completely out of context, you would say that that patient has metastatic disease as in not responding to that's not me, not responding to treatment, but in this particular context, she still takes 1 dose a day and is fine. We also, when the patients became resistant to imatinib and we started sunitinib, we decided to, somebody has a phone ringing, we use the same principle of doing FDG at baseline, and you can see this patient has almost his entire liver involved, an exophytic mass in the abdomen and some other masses here. We imaged that patient early after the initiation of the novel drug, and you can see again complete inhibition of almost all of the glycolytic metabolism. Now, in this particular trial design, the patient then went on what's called a drug holiday for 2 weeks to look for toxicity. And what we decided to do is to image that patient at the end of the drug holiday when the patient was off the drug. And you can see that there is re-emergence of glycolytic activity, glucose metabolism, in the masses that the patient presented before. But when the patient is back on the drug is, again, hitting the target and controlling that tumor, and the patient remains without any evidence of disease on a metabolic point of view in subsequent cycles. So what we learned from that history is that imaging can provide proof of concept for target modulation. It can help test the underlying hypothesis. It can inform rational selection of dose and schedule, which it did in this happened to then become Suitent, and aided decision-making, including key go, no-go decisions. And the drug, more importantly, reached the market 6 months ahead of schedule. And just from private communication from Tim McCarty, this is a Pfizer drug, the estimated savings for the company were $200 million plus sales. I'm still waiting for a penny on each one of those dollars, but I'm still hoping. But for you in your in drug companies, this may be an important point to remember. Another way to address it, this was the very first CDK4/6 inhibitor trial. And we did it in mantle cell lymphoma. This is before breast. And we decided, because it was a thymidine kinase inhibitor, to use fluorothymidine, which can look at the proliferation, and then repeat that after 1 cycle. And you can see that at baseline, there's tremendous proliferation within all the sites. You see all the adenopathies within the body are extremely in full reproduction, but the drug hits the target and decreases that. This was confirmed by Ki67 inhibition and phospho-Rb, and you can see that most of the patients showed a significant decrease in proliferation with that methodology, and that was actually predictive of progression-free survival. The patients who showed the response were doing better. However, in that trial, I also wanted to add, as a control, the routine PET that we do, which is FDG PET, looking at glucose metabolism. And so here's a patient with the FLT PET on the left at baseline after 1 cycle. And here is the FDG PET at baseline and after 1 cycle. And you can see that there is absolutely no difference here between the pre and the post. So despite the fact that the target was hit, and the patient showed a significant decrease, 43% after 1 cycle, that did not translate to modifying the metabolism on which the cancer is relying to survive, in this case, glucose metabolism. And these patients in the trial actually showed progression. So using two ways to interrogate the tumor, both in the microenvironment and specifically in the target, helps a lot. And this paper I thought was interesting because it showed that the probability of success for any of the phases 1, 2, 3 or whatever is actually augmented if you add a selected biomarker. And you can see that every single phase has benefited from having a patient selection biomarker. And then, the likelihood of success is also higher with it rather than not having it available. And so one way to also improve the translation is to maybe provide a concept that we adopted, which is called the co-clinical trial. When I became chair at the DANA, I was offered an opportunity to ask for what I wanted, the wish list. So I asked for a translational imaging site that we call the Center for Biomedical Imaging in Oncology. So we got funding to create the preclinical imaging facility, which is we actually on the harbor. If you want to visit us, everybody's welcome. We share the space with the cruise terminal, so you go as far as you can, and you find us. And then I had to wait 5 years, but I was able to rent some space next to the facility to then add a cyclotron, and we now have a highly active molecular cancer imaging facility that does a lot of R&D support for academic investigators as well as industry. And all the imaging is behind the barrier, so we never have to kill an animal. We can actually test an animal the same way longitudinally with repeat imaging as we would do for humans. And that really has helped actually transition some of the drugs much faster into the humans, and was actually used to change the recruitment in some of those trials, because the information we got on the preclinical side, for example, in GEMS, in 3 different GEMS models, showed that one, and we did this with a MEK inhibitor, one of the mutations did not respond at all. That mutation was not tested in the humans. The clinician studied, who was running that trial at the time, started to test patients, excluded the patients with that mutation from the trial, and suddenly his response rate went way up. And this ended up being selumetinib at the end. So I think that we have ways to actually try to shrink that time period. And I mentioned the fact that we need to assess the TME. I just have time to just show you, and people have mentioned, FAPI is very popular right now. Cancer-associated fibroblasts are the most abundant cell type in the TME. It makes a lot of sense to target them. They have poor prognosis, and they express a particular protein that we can target with an imaging agent as well. And so far, it has been seen already in at least 28 tumor types. So it's not specific to one particular tissue. The tissue of origin becomes irrelevant nowadays. You can really look at, again, across many solid tumors as shown here. I don't have room to put 28, and it's growing. So pan-cancer ability, the same way immunotherapy is pan-cancer, meaning the tissue of origin is irrelevant. You can actually treat many solid tumors as well as liquid tumors, is really something exciting to consider. And it has moved into the radiotherapeutic era. Here's an example of a gallium FAP image showing tremendous, the entire bone marrow in this patient is actually involved following injection of lutetium FAP, and a significant response already. Another one that is of interest is to look at the immune system. We know that the tumor is able to block the immune reaction. Can we interrogate that immune environment? And there's now ways to label various cells. I'm just going to show you CD8 as an example, because one thing that is being asked for people who are candidates for immunotherapy is to us is to say, can you help me take the temperature, as they call it, of the tumor? And this is a question that Aline Sharp, who is the chair of the Department of Immunology at Harvard, asked me at the time when she was presenting, doing a presentation, and because she clearly said there are 3 main anti-cancer immunity phenotypes in humans, and they're critical for predicting if a patient will respond to treatment or not. And one of them is shown here, and you can see all those brown spots are actually T cells. The line shows the boundary between the tumor and the microenvironment. around it. And you can see that not only is the patient able to activate these T cells, but send it to the tumor and inside the tumor. This patient also is able to make an army of activated T cells to go and fight the tumor, but the tumor prevents the entry. It's like a castle with a moat around it. And then patients may not show any evidence, and they're called immune hot inflamed, excluded, and cold desert. So if we have a way to actually do that, and I'm just going to focus on this patient here. This patient has a metastatic lesion in the calf here. It has melanoma. This is what the FDG PET looks like. This is the CD8 PET. So you can see that prior to any treatment, there is evidence of activated T cells there. The patient then goes on to one cycle, and 1 month after treatment, you can see that the CD8 PET has increased markedly, while the FDG PET has actually decreased. And you can see the cross-section imaging here showing the significant, first of all, the presence of CD8 prior to, and then the marked increase. And I think that I'm going to finish on this because I think there is a piece of news, a good news in the epochal transformational history of cancer we've gone through for almost a century now, is what Arlene Sharp is referring to as the Kaplan-Meier curve. As you know, we actually following success by asking if the patient is alive or not. And with chemotherapy, we have made some progress, and we have increased the survival with the introduction of molecularly targeted therapy. We pushed that even further, but none of these actually show durable response because eventually the patient passes away. But with the very first trial of ipilimumab in patients with melanoma, we started to see what she refers to as a flattening of the curve. And that actually has been recently confirmed with the update on the data in that trial. This is the ipilimumab trial. Now, we are now past 10 years, and you can see that that flattening of the curve has maintained itself. Moreover, with the introduction of PD-1 nivolumab, we actually have elevated the survival to just below 50%. And if you combine both, we're actually above the median survival. So the hope is that we are going to raise a level where we can actually provide a durable response to most patients, and wouldn't that be the best game changer of all? So I know I have a few more slides, but we need time, so I can stop here.
I will do the introduction. The last speaker.
Oh, okay. So
I'm so sorry.
If that's all right.
Sorry about that.
Yeah. Well, collaboration and partnership so important. So my great pleasure to introduce Cheryl Massisian. She's got the PhD in physics. She's the Chief Scientific Officer at SOFI Biosciences, and she'll be talking about probing potential, how to find success, leveraging academic biotech partnerships.
Thank you very much, Ilya. Appreciate that. The last talk, we're almost there. One of the great things about being last is I get to leverage all the great talks given by the previous speakers, so thank you for that. Barbara, thank you for this amazing opportunity. It's absolutely incredible, the work that you have done, and truly a pleasure being here, so thank you. There we go. I won't spend too much time on this piece, as I know the previous speakers have done a tremendous job of it. There's one portion that I do want to highlight, and I know Dr. Abele touched on this, which is a timeline for activation. When we look back at the tremendous success that came from Dr. Saul Hertz, and in founding this field, it took about 10 years from proof of concept to the approval by the FDA. We fast forward, we've seen incredible success over the last couple of decades that has led to the current explosion of the field. Having said that, the timelines are still quite slow. We're looking at 20 plus years to the approval process. And here we just break down the very general concept of what it takes to go from discovery all the way to market access. And this applies to any drug, of course, but we are focusing on radiopharmaceuticals. So one piece is discovery. which is really looking at a variety of products and looking at the best ones that come to the top. Proof of concept. For radiopharmaceuticals, we have a great advantage where proof of concept is very much tied to discovery, and the bar to test in humans is low, with great potential. We have regions of the world, Germany, for example, Australia, that have a very streamlined pathway for first-in-human results, which again gives us a lot of advantage. And then we have the processes of scalable manufacturing, logistics, clinical trials, and regulatory execution, phase 3 studies, especially towards approvals, and lastly, market access. Now, we don't have to divide this up by what entity does best in this, but there are trends. So when we look at this, we see academia does an incredible job with discovery. And due to flexible regulatory requirements in certain regions, for example, compassionate use, allows for the testing of the proof of concept to be done quite effectively. The next stage where we're going to spend a little bit of time talking about this is the biotech small pharma concept. And one of the advantages of biotech small pharma is the ability to be a little bit more risk tolerant, have nimbleness in moving things forward, while still being able to structure processes that are needed for large-scale manufacturing, for launching prospective clinical trials, engaging with regulatory entities towards success. And last but not least, we have large pharma. Again, not to say large pharma would not be good at the other components, but large pharma has a higher or lower threshold for risk, but they're very good at putting in the mechanics of what it takes to bring a product to market. So let's look at what we've done over the last major success stories in the field. So when we look at the somatostatin receptor successes, the pathway started, of course, with academic partnerships. We saw AAA license the IP. We had pivotal phase 3 studies, larger scale GMP production, labeling kits making routine hospital use feasible. NetSpot got approval in 2016. Lutathera got approval in 2018 by FDA. So we see here about a 25 or so year gap from concept to eventual approval. Novartis acquired a AAA in 2018. The next example is Pylarify. We're using Pylarify because that was the first PSMA diagnostic. Again, 1990s, Dr. Marty Pomper's group at Johns Hopkins developed the product. They had done proof of concept studies in humans, and the product was licensed by Progenics. Phase 3 trials were ran. Again, GMP capabilities were expanded, NDA submission. 2021, it was approved. The company was acquired by Lantheus in 2020. And then we have Pluvicto, PSMA binding for therapy. Here, again, very similar story. Heidelberg University had done the initial compassionate use studies with the PSMA therapy. Endocyte licensed the IP. Vision trial took place for phase 3, again, along with global manufacturing, supply chain logistics. And Endocyte was acquired by Novartis in 2018, and then the commercial launch took place. I'm so glad Dr. Abele went before me. It could not have set up my talk in a better way. So we're seeing these great success stories, the role that smaller pharma biotech plays in, I call it as an accelerator. We take something that's really promising, put more structure behind it, and make it a reality closer to bringing it to the patients. What is next? Again, I might be a little bit biased considering that I work on FAP, but looking at all the publications and the products that we see, fibroblast activation protein is being seen as a very promising target. It is a very different story than PSMA, but it does have incredible potential, almost too much potential. So again, Dr. Abele covered a lot of this. I'm not going to go into too much detail. Two important factors is FAP is expressed in activated fibroblasts. So when we're looking at the potential of FAP, there is oncology, and then there's non-oncology that should be looked at. Oncology, of course, we're looking at the CAFs that are in the tumor microenvironment, which again is a great opportunity to target the cancer cells based on a TME, but at the same time, as Dr. Abele also mentioned, it becomes increasingly important to look at both the expression of targets at the tumor cells in addition to the tumor microenvironment. Now, when we're looking at indication potential for FAP, this is where the challenge lies. So non-oncology, there are a variety of possibilities. We have arthritis, cardiovascular disease, liver fibrosis, lung fibrosis, Crohn's disease, IgG4 disease, anything really fibroinflammatory could be a great potential for the product. And then we have oncology. Close to 30 different oncologic diseases have been imaged with FAP quite successfully. 90% of cancer is highly expressing FAP. So the possibilities are quite large. And GI tract, cancer of the GI tract, so gastric, esophageal, colorectal, hepatobiliary, sarcomas, bladder, cancer of unknown primary, subset of lung, subset of breast, ovarian, and more. So this is a great story. The challenge is this would equate to about 30 different prospective studies to explore, which would be quite challenging to do. So the approach that Sophie's taken, and now we are collaborating with GE Healthcare on this, is to, again, looking at this diagram, and it's important to note that the academic biotech partnership is not a one-way partnership. This is an iterative process that feeds into itself. So what we have done is we have taken this asset that has a lot of potential, and we have asked for help. We've asked academics to do what they do best, which is asking great questions, being innovative, looking at possibilities of indications. We provide them access to GMP-grade precursor. We give them manufacturing and regulatory support, and this is their study. We review the studies, and if the studies are appropriate, we sign off on it, and then they run their own study. 70% of these are clinical studies, about 30% are chemistry and preclinical. So far, again, our team has led an incredible effort on this. We have 41 countries represented with FAP studies. Over 380 research studies are taking place in oncologic and non-oncologic disease across 169 unique institutions. So what is the value of this? Well, to academics, there is tremendous value in academic curiosity, securing grants, reputation through publications, and being able to advance the field. For Sophie and also GE Healthcare, what is the advantage? The advantage is learning, seeing the proof of concept data. So we're not guessing as to what is the next potential that can be successful. So has that resulted in success? The answer is yes. We're being very systematic about what we should work on and where we should launch prospective studies. As I mentioned, we would not be able to run 20, 30 prospective studies. That would be very limiting. But what we have done is we look at the data that has come from the Global Outreach Program. We have looked at our own phase 2 study, which was in GI cancers, and we have worked closely with the FDA to take the next step in bringing FAP as a diagnostic to the clinic. So the phase 3 studies are highlighted here. These will be launched in about 3 to 4 weeks. FDA has already given the green light, and these are 2 studies. We will have 17 sites across the US to run both of these studies. We believe that metastatic disease detection is where FAP does really well. So the focus is detection of M1 disease in gastric and esophageal cancer, and a separate study in detection of M1 disease in PDAC. Is this the end of the story? No, this is just the beginning. Our goal is to, again, go back to that relationship with academia as we launch these 2 phase 3 studies, go back, look at the proof of concept data, and pick the next promising indications to bring into this pipeline. So again, that iterative process, close collaboration with academic and industry partners, to us is the best way to bring this very promising target to reality. Last but not least, this takes a lot of effort. Sophie's a smaller company, but we have about 550 staff across the network. We have a radiopharmacy network, which is a massive asset to the work that I do because I get to feed the radiopharmaceutical into the network, and we have 15 sites across the US that can support our studies. Our clinical team, our scientific medical advisory, Dr. Shadi Esfahani, right there in the middle up top. She's been an amazing asset. She's our colleague at MGH who led the phase 2 study. MGH got 50% of the patients for the study came from MGH. So it was a tremendous success story, and we're looking forward to continue the collaborations. And that is the end. Thank you. I'd like to introduce Alex Brown, COO at Convergent Therapeutics, to be moderating the panel.
Yeah.
I gave you more than 5 minutes.
You gave me more than 5 minutes. Thank you.
You're welcome.
So I don't have any slides. When Barbara asked me to participate, she gave me the option of doing a presentation or asking really difficult questions to the panel and the audience. So I went for the first. I have a lot of difficult questions, but I think we only have about 5 minutes left. So we're going to ask the panelists to come up and at least address one question I prepared, and then see if there's anything that the audience would like to ask as well. All right. So probably the easiest question to ask, but the most difficult to contemplate. We heard a lot about how the folks here today have built on the legacy of Dr. Hertz in their own work or in the work of their institutions or their companies. And I think the question I would pose is, where do we go from here? And in particular, how do each of our panelists see the opportunities or the barriers that we need to overcome or pursue to continue this legacy of Dr. Hertz? So I'll begin with Professor Langer and what your thoughts are.
Yeah, well, I think about this quite broadly because what Saul did was incredibly broad. Sure. Thank you. Is it on? Is it on? Yeah. Okay, great. Yeah, I think about this quite broadly because I feel what he did was extremely broad. But I mean, one obvious area I think that is like targeted radiopharmaceuticals and, to specific cells, cancer cells, all kinds of things. So I think, I think it really, I think there's so many directions you can take it, but that's certainly one of the areas that I think would be very important.
Yeah.
I focused in my talk exactly on the challenges. So all I'm going to say is that that was an extremely important first step of the journey that Dr. Saul did. I think that some of the challenges that we face, and some of those that I verbalized during my short speech, is because we are now having the potential to solve them. Those computers, those AIs, those models that we can bring into the field of radiotheranostics are opening new horizons, and those challenges would not exist 50, 60 years ago. So I think we're well positioned to solve problems of additional personalization, individualization, more clinical applications, and I think that is just so exciting.
From my point of view in neurodegeneration, we are thinking about earlier, and earlier. We accumulate some tau protein as part of the normal aging process, but where does that synergy with amyloid, where, again, I mentioned before, where is the tipping point? How can we better understand how to address this disease earlier? And again, the importance of development of alpha-synuclein PET tracers to contribute to dementia with Lewy bodies, Parkinson's disease, trying to help contribute to therapy development for other neurological disorders.
Where do I start? There are several things that I get a little bit ticked off about when it comes to novel therapies, and I read the manuscripts, is the fact that they often, and I think that's the case with FAPI versus the FDG that we use in the clinic every day, where the paper say this is better than. But I think that there is a lack of understanding really of what the biology is. And I think it's inappropriate really to compare two things that actually interrogate a particular tissue or tumor in two different ways. It's apple and oranges. Is the apple better than the orange? That depends on your taste, right? I mean, the data do not prove that you should then switch your practice and suddenly drop something you have been using and have a lot of experience with. You always have to put it in the context of the biology. And I think that David brought a very good point about education and about the fact, one slide I didn't show is the fact that we have really dim news, coming up. There's going to be between now and 2050. Do you know what the statistics are when it comes to the increase of cancer? It doesn't matter which continent you live in, it's going to be 70%. 70. Can you believe? I mean, the amount of cancer, that's probably also related to the fact that we can diagnose it earlier, we can, recognize it quickly, but this is where it is. I think I feel similarly to what Julie was saying when it comes to Alzheimer's, because it is also going to be something jumping at us. So we need to be prepared to address that. We need to educate and understand what we are treating, what we are imaging, in order to make the right decision for patients. And then the third point is what I brought up, is the translation when it comes to going through the clinical trials and getting to the patients. I think that particular area needs to be addressed. It's funny that you mentioned the FAB right now, and I'm looking at your phase 3 trials. One thing that we thought was going to be very simple many years ago when we were fighting to get FDG approved by the FDA and by CMS in order to get reimbursed for it, is that we'd said, well, that's a common metabolism, against all tumors. So we want a pan authorization. It doesn't matter what cancer you have, you should approve it for all of the cancers. We can prove there is an increased glucose metabolism. There was a complete lack of understanding of the biology at the time. And, we always used to look at CMS and FDA as being the Everest. I call them the Mount Everest. Impossible to move, they're those giant mountains. At the end of the day, there are very few people in those groups having to make a decision for millions of people. And I think that you need to be educated. And I came to the realization, and that changed the field completely when I started to understand what they needed to learn, and then they needed to teach me how they go about the decision, what does it take, why did they say no, why this, why that, and that facilitated the communication tremendously. So I think communication and education is really important. Sorry.
Oh, okay, we're about to lose the room, so I'll be focused. For opportunities, we've already heard that from the various speakers that appropriate use is a very important focus. The right patient, the right dose, the right timing. And for challenges, we've come up with various catchphrases in the company, which I think people tend to do. And one of the catchphrases we come up with as we move from one target to another is that we know nothing. We just have to start again. We reconsider that target's biology and how best to address it for the benefit of the patient. So that probing sort of openness, intellectual curiosity that sparked Dr. Hertz to ask if radioiodine could be produced is how various different companies approach these problems, but it's a problem that can also be addressed best by the intellectual openness and curiosity of academia, at which point I can hand you the microphone.
Thank you very much. As I was coming to this conference, I was rereading the story of Saul Hertz, and I thought I knew the story, but I didn't. And I think a couple of things that came obvious to me is there's a level of tenacity and a level of perseverance that was pretty obvious. And I think my hope for our field is there's been a lot of investment coming into our field, which I think is tremendous, especially from large pharma. That is good, but that can also be challenging. Because I think sometimes it takes away some of the creativity, some of that perseverance, some of that tenacity. I think funding should really be much more provided to academics where the creativity sits. So I think for us to not lose that tenacity and not become so mainstream to the point that we are just all doing the same thing over and over again for the next target, to get to the market, that would be very boring. And I would have no doubt that based on the audience and the creativities, we won't have that be the problem. But I think we have to be very thoughtful about not falling into the comfort zone. Thank you, Barbara.
Thank you, Barbara.